This model recapitulates both the pre-malignant and tumor microenvironments by incorporating physiologically relevant hydrogels with tunable stiffness and hepatocellular and stroma-associated cell lines.
This model is modular and cost-effective, can be prepared with basic equipment and readily available materials, and can be used to study complex tumor-stroma interactions.
This tumor-stroma interaction model can provide insight into the hepatocarcinogenesis, and this is very important for both a mechanistic perspective and a treatment perspective. Demonstrating the procedure with Carlemi Calitz will be Jenny Rosenquist, a doctoral student from the Ångström laboratory.
To prepare 10 milliliters of an 80 milligram per milliliter fibrinogen stock solution, first add 2.21 grams of calcium chloride and 5 milligrams of aprotinin to individual 20 milliliter volumes of distilled water. Then add 2.051 milliliters of the aprotinin stock solution and 100 microliters of the calcium chloride stock solution to 7.849 milliliters of PBS. Next, incrementally add 800 milligrams of fibrinogen and 200 milligrams of sodium chloride to the solution without stirring or shaking, and place the tube of fibrinogen stock solution horizontally on a shaker. After 2 to 5 hours of shaking at 300 revolutions per minute, filter the resulting solution to remove any clumps that formed during the preparation. For cell culture insert collagen coating, in a biosafety cabinet, use sterilized tweezers to invert the inserts onto the lid of the cell culture plate.